All right, good morning, everyone. I think we'll slowly get started here. There's still a lot of people joining, but in the interest of staying on time, I wanted to welcome everyone to the third of three BioGeoSCAPES modeling webinars. And just to kind of get everyone on the same page, what is BioGeoScapes? Well, it's this global initiative. It launched kind of mid 2020s, and the aim is to advance our understanding of ocean microbial biogeochemistry. And there are kind of three main pillars of BioGeoScapes: states, mapping key ocean metabolisms over space and time; rates, mechanisms connecting microbial metabolisms and productivity to biogeochemical cycles; and fates, predicting interactions with environmental factors to provide risks and vulnerability assessments for key ocean metabolisms. And a key goal of BioGeoScapes is to create this interdisciplinary international group. And one of the aspects of that is to engage ecosystem models and other types of models and omics data and experiments and observations. So BioGeoScapes has multiple different scales it looks after, and that mirrors what we've been trying to do in this modeling webinar. So we think both about intercellular, cellular scale processes, how that translates into understanding organisms and microbial communities, and then how that scales up to the global scale cycles and ecosystem scale feedbacks. For the goals of the modeling webinar in particular, we really wanted to create a platform where we could connect modelers of different types of techniques and all computational scientists working on these issues. And so the idea is to highlight the diversity of types of models that are being used to assess BioGeoScapes type questions, and to try to bring our community together to understand how the sum can be greater than the parts. And the ideal outcome of both these webinars and in the in-person workshop will be to try to increase collaboration actually between modelers and potentially create new frameworks that we can use to address BioGeoScapes questions. So as I mentioned, this is a third of three. If you've missed the other two, they're online. And you can find them by going to the BioGeoScapes website. And the first one was on biogeochemical models. The second was on network and statistics, and this one will be on cellular scale processes. It's been super exciting to see the interest in these webinars that we've had over 261 registrants, and these are the distribution of countries, so really a global interest, which is super exciting. And we've had a lot of fantastic attendance at all three webinars. And so then the next phase will be an in-person workshop. That registration is going live as of now. It will be held in September in Woods Hole. And the applications you can find, you will get an email that will go out to the list for people registered for the webinar. And you can also find it on the BioGeoScapes website or this QR code here. And it will be open for about a month, closing on June 30th. So today we've got two amazing speakers. We've got Dr. Suzana Leles, who's going to be starting at faculty at UT Austin. And we've got Daniel Segre from Boston University. And we'll have, just like the other two webinars, 20-minute talks in sequence. And so the question A will be at the end of both talks. So please submit your question to the Q&A form, and we will have a discussion at the end of both talks. And just to remind everyone before we get to the actual science, why is it not advancing? I apologize. There we go. We have a code of conduct, and it's really important to us that everyone feels included and has a voice. And so we do not tolerate any form of harassment, bullying, intimidation, or discrimination. And so really make sure that this is an open space, both the webinars and the in-person workshop, for people to come and share their ideas. So please be respectful. We can critique ideas, but not individuals. And if you see anything that you feel is troubling, really please report it either through the QR code or emailing one of us, the panelists or the organizers. So with that, I am super excited to kick off our third modeling webinar with Dr. Suzana Leles.
Thank you, Naomi, for the introduction. Let me share my screen. All right. Awesome. Can you see it? Yes.
And so it looks great.
Awesome. Hello, everyone. I am super excited to be here today. I've been following this webinar series, and it has been very inspirational to me. And today, I hope to contribute with my perspectives on cellular scale models, and especially how we can integrate this type of models to different data sets and to other modeling approaches. And to do that, I will share with you a case study on how we apply cellular scale models to gain new insight into phytoplankton biogeography. But before diving into this story, let's look at the big picture and also talk a little bit about what are these cellular scale models. So as we know, marine microbes, they act as biogeochemical engines. Phytoplankton, for example, they are responsible for producing half of the oxygen on Earth. And remote sensing tools give us this incredible ability to actually map chlorophyll concentrations across the globe. However, this map that we are looking at and also other large-scale processes that we observe, they are being driven by cellular level processes. And with the advent of the molecular revolution, we were actually able to tackle microbial function and biogeography by looking at these small scales. Here, for example, you can see a map of the total metagenomic sequencing effort across the ocean to date. So these allowed us to tackle questions that perhaps before were hard to do without this information at the cellular level. So we can now go deeper into the mechanisms behind ocean biogeochemistry. And in parallel to these advancements, we also see advances in numerical modeling, so we can represent more complex microbial communities to investigate their impact in biogeochemical cycling. While these approaches, they are very powerful, the challenge for now as a community is to integrate these different approaches or to apply them in a synergistic way so that we can really go from the small to the large scale. Personally, on my work, I think a lot about how we can apply these cellular scale models to better understand trade-offs that microbial cells face under different conditions. And these models, they can be very powerful because they offer a way to link numerical modeling to the omics toolbox, but also they work like a bridge between omics data sets and biogeochemical models. But you might be wondering now, what exactly is a cellular scale model? So let's look into that into more detail. So modeling cellular scales is all about allocation and trade-offs. And let's take time as an example. Time is a very valuable resource to all of us because it is finite. And we need to choose how we allocate our time during the day. So let's say our objective is to maximize time with our kids, but we have a proposal deadline approaching, and we also must pay our bills. So that means that if we allocate time to one function, we cannot allocate time for another function. So we can spend some time with our kids, but we also need to work. And this leads to trade-offs. So we can apply the same logic to microbial cells. So microbial cells, they must acquire resources, which can be like energy, carbon, or nitrogen, or other nutrients, and they need to choose how they're going to allocate these into different functions. So for example, we can think that the objective of our model here is that a cell should maximize its growth rate under resource limitation, but also pay for the metabolic costs. This is just one example, but actually, according to your question, you can define different objectives. So for example, you might be interested in maximizing energy use efficiency, energy gain versus energy spent. Or perhaps you must minimize a certain property, which could be minimizing total energetic costs. So it really depends on your question. You can change this objective function. But overall, these models, they share like a common structure. So in order to find the optimal metabolic strategies of a microbial cell, we need to start formulating our model, choosing the components that we are representing, how they interact, what are the constraints, what is the environment. We make the assumption of steady state and also mass balance conservation. We define the objective function, and we choose an appropriate optimization technique to find our optimal solutions. Now, this is a very broad overview, and I wanted to dive in a little bit into different models that we see to actually tackle these questions at cellular scales. And by no means this is a comprehensive review, but I decided to focus on three types of models that vary in the degree of the metabolic detail that they can represent, and also how they can link to omics data and to larger scale models. So let's start with the macromolecular model. So here we have a model that represents a very few number of pools. So it's the most coarse version, let's say, but it has the advantage that optimal solutions can be derived mathematically. And this really makes these models more easily integrated into biogeochemical models. At an intermediate level of complexity, we have protein models in which we typically represent more metabolic pathways. We have nonlinear constraints. It's more mechanistic, but it also comes at the cost of more parameters. But the interesting feature is that there are many opportunities to leverage omics datasets with this type of models. And finally, on the other side of the spectrum, we have metabolic flux models, which really provides a fine resolution of metabolism, focusing on fluxes, makes less assumptions about metabolic dynamics, and rely on metabolic reconstruction based on annotated genomes. Now, this is not my expertise, but don't worry, because Daniel Segre will be coming just after me, sharing his perspective on this, and I'm excited to hear that. So I am more familiar, what I use for my work is these two type of models. And today specifically, I will be focusing on proteome models. So as a case study for today, I will present our work where we applied a proteome allocation model to investigate phytoplankton biogeography under future conditions. And as a teaser, this story has a lot to do with cell size, and our model can actually predict increases or decreases in cell size as a function of the environment. So as a quick intro to phytoplankton biogeography, we know that there are well-known distribution patterns for different types across the ocean. So for example, the tiniest of the phytoplankton, cyanobacteria, they dominate within oligotrophic gyres. And larger eukaryotes, such as diatoms, they will dominate in more nutrient-rich regions. However, if we think about what is happening as the climate changes, we know that as the climate warms, current projections, they predict an expansion of the gyres, which therefore results in an expansion of the niche of cyanobacteria, especially Prochlorococcus. And they are expected to be more abundant under future conditions. If we think about what is happening at the cellular level, we can imagine that as the climate warms, nutrient limitation can be more severe in surface waters in the ocean. And in order to optimize resource acquisition, a phytoplankton cell should invest in transporters, which requires larger surface area to volume ratios, and therefore smaller cell sizes. So this is a thumbs up for why cyanobacteria should also predominate under future conditions. However, if we take the multi-stressor condition, they also must handle heat stress. And while you might think that phytoplankton will become smaller as temperatures increase, this is not the case when temperatures are critically high and they need to handle heat damage. Under these conditions, phytoplankton must invest in heat mitigation strategies, which mean accommodating for antioxidants and also energy reserves. So cells must actually get larger to handle heat stress. So if you are curious about this, I invite you to check our past work where we describe these mechanisms in more detail. But for today, I hope you can believe me that there is a trade-off here. So under the multi-stressor condition, we have cells that must optimize nutrient acquisition by becoming smaller, but also optimize heat mitigation by becoming larger. And a question arises from what is going to happen under future conditions, because our current models, they represent phytoplankton physiology in a very simplified way. And we don't account for heat mitigation strategies or carbon storage. So the question is there a niche for being larger in the ocean, or being small is going to always be advantageous? In order to tackle this question, we apply our single-cell protein model for phytoplankton. And here you can see the different components that we have in the model. In blue, we have the proteins, and in green, we have other macromolecules like lipids. And the arrows show how these different pools interact. So basically, with this model, we can actually find the optimal metabolic strategy of a given phytoplankton cell that will maximize the growth rate. And by doing that, we can investigate shifts in the relative proteome investments into different functions as a function of the environment. And as a reminder, our model can predict the optimal cell size that will maximize growth. So what we did next, we investigated phytoplankton strategies to combine heat and nutrient stress. And to do that, we solved the proteome model across a range of temperatures, different nitrate concentrations, and also we varied key model traits to represent a diversity of phytoplankton phenotypes. So for each phenotype, we could get something like this out of our model. So this is an emergent property of our model, and we can see that we can simulate thermal growth curves under both nitrogen replete and limited conditions. But what was interesting is that by simulating 100 phenotypes, we identified two key strategies. So the first one, the nitrogen efficient that you can see on the left, represents cells that to handle multi-stressor growth, they chose to maximize nutrient uptake efficiency and growth, and therefore they would require smaller cells. And on the other side, on the right, we have the thermotolerant strategy, which allows cells to survive at higher maximum temperatures at the cost of lower maximum growth rates and also requiring larger cells. So we can relate these two competing strategies to these two phytoplankton functional types that we were talking before. So cyanobacteria are optimized for nitrogen uptake, while larger eukaryotes, such as diatoms, they do better at mitigating heat stress. What we did next was to actually calibrate our model for these different types. And we used empirical thermal growth data to do that from a previous compilation from Anderson and collaborators. And this work was done in collaboration with Dr. Ariana Krinas. So we were able to use data for over 160 species to calibrate the protein model. We then validate our prediction against trait data. And I apologize for not having time to go over all these. But after doing this validation, we also worked with Dr. Maria Aranguren-Gasses to validate our modeling predictions against a proteomics data set. So we can do that in a qualitative way, where our model tells us whether a pathway was up or downregulated from environment A to environment B, and we can compare that against the omics data set. And while this was done qualitative for this work, I invite you to chat further with Ariana Krinas, which is applying this model in a more quantitative way with transcriptomic data sets. So finally, our last step was to use, to team up with Earth System modelers, Lana Flanjak, who is a PhD student working with Charlotte Lovecotter, and we solved the proteome model for present and future projections of temperature and nitrogen concentrations across a latitudinal transect in the Atlantic Ocean. Here is just one example of the sort of output that we get from the model. So we plotted here the optimal growth rates across this latitudinal transect for our different modeled phenotypes. But I want to focus on one key finding from this study. So we found that in the poles, all three phenotypes, they optimized it to be smaller under future conditions because nitrogen stress was more severe than heat stress. However, we saw the opposite pattern in the tropics. In the tropics, heat stress was so strong that cells were actually driven to get larger. They had to invest in heat mitigation strategies to handle this condition. And what this resulted in is that we could see an expansion of the cyanobacteria niche from present to future conditions in temperate to polar seas, but this was not observed in the tropics, where cyanobacteria were more vulnerable because they have lower heat mitigation capacity and they are also smaller. So what our study suggests is that perhaps there is a niche for being larger in a future ocean, which could be favorable for Synechococcus or even larger eukaryotes such as diatoms. Having said that, I think I am on time. And I wanted to acknowledge my postdoc advisors, Naomi Levine and Holly Moeller, and all the collaborators for this work. It was really a team effort. And also acknowledge the lab members from the Levine lab and the Moeller lab for feedback in previous versions of this work, and our funding agencies. And I will take 2 seconds to just advertise that I am starting my own lab. It will be effective in August this year at the University of Texas at Austin. And I'm hiring grad students and postdocs. So if you're excited about these sort of questions, please don't hesitate to contact me. I'm excited to meeting you. Thank you all for listening.
Thank you, Suzana, for a great talk. And we're going to jump right into Daniel's talk. But again, please put your questions for Suzana and for Daniel in the Q&A, and we're going to have time for a discussion at the end with both Suzana and Daniel. Looks great. Yeah.
Okay. Can you see and hear me?
Yes, excellent.
Great. Thank you very much for the opportunity to talk to all of you. Great to connect to this community. I First of all, I want to say I'm grateful to a lot of people. The reason I'm studying marine microbes is really thanks to a lot of people that I've encountered along the way. Most grateful to the collaboration with Daniel Scher and Spetter that started me on this avenue, and recently the CCOM Center for Chemical Currency of a Microbial Planet, NSF Center led by Marianne and Liz, and many, many people that really contribute to the research we do. So I've been thinking about metabolism for a long time in different ways. And overall, if I had to summarize what fascinates me the most about metabolism is really this multi-scale nature that many people before have pointed out in this series. I find particularly interesting that somehow this very same molecules that we can study in a cell, we can study also at the global scale, and that somehow we can go up and down this hierarchy. So I think models don't necessarily need to just go up from smaller scale to larger scale. And flux balance model, as I'll mention, is one example where knowing something about the what a cell do will inform also how reactions operate. And in the lab, we've been recently particularly interested in exchange of nitrogen and carbon between heterotrophic bacteria and communities of microbial heterotrophs, both in association with plants and with phytoplankton in the oceans. So one of the big questions we address using modeling is how to understand the way microbial metabolism in individual microbes determine ecosystem function and dynamics. And I illustrate this in 3 possible ways one can address this question. And there are models where individual microbes are kind of black boxes and there is no real internal structured information. There are more detailed models where one can try and understand how ecological dynamics and interactions are determined by intracellular circuits. And I think it's also interesting to think of the possibility of modeling metabolism as a soup of enzymes where we may not even take into account what organism perform what functions. And maybe that's something we can discuss later. But I'm going to focus on this type of modeling, very much building on what Suzana just showed you. We're very interested in trying to figure out how intracellular wiring in individual organisms and environmental constraints determine ecological interactions, dynamics of microbial ecosystems, and whether we can understand the systems well enough to potentially design communities with specific functions and purposes. So this is how I illustrate the model, and it's again a problem of resource allocation. And what I find fascinating, right? Every single cell solves this problem all the time, constantly, nonstop. This is just a very rudimentary representation of metabolism in heterotrophic like bacterium. You can recognize here glycolysis generating some ATP going from glucose to pyruvate. There is a mock respiration that generates even more ATP with oxygen coming in. And these two reactions represent the production of biomass components. Here are arbitrary molecules A and B that cause some ATP. And the reason I'm showing this is just to illustrate the fact that if I ask you to find one set of reaction rates that satisfy all the conservation constraints of the mass of each of these metabolites, for example, just even asking that the ATP that is produced matches the ATP that is consumed, it would be very hard to find a solution, to find a set of numbers to put on these arrows. And this is why we need mathematical models to address metabolic questions. And we and cells care about the rates of metabolic reactions, of course, for understanding biogeochemical cycles. The secretion of specific compounds can mediate interaction between organisms. By knowing the rate at which biomass of different organisms are produced, we can estimate community composition, and so on. So, if we don't understand, if we don't know how to estimate rates for a simple model like this one, how are we going to do it for real cells, which have of the order of 10 to the 3, about 1,000 reactions? And that's where this idea of stoichiometric modeling comes in, as you heard from Suzana. I want to illustrate this with a very simple example, just to point out a few things that I think are really important. So this is a tiny reaction network with 3 fluxes. And if we wanted to model this with kinetics, with chemical kinetics, each of these fluxes, right, would be traditional Michaelis-Menten equation, probably more complicated than this one. But what is important is that each of these rates does depend on the substrate concentration, of the enzyme concentration, and kinetic parameters that are typically very difficult to measure. So when you have 1,000 reactions, each of them having a Michaelis-Menten term like this, it would be really, really difficult, and it is challenging, not impossible, but very challenging to build models that can predict the set of fluxes in a whole cell. But the steady state assumption we heard about before is what makes this problem tractable. And Suzana was talking about time. I'm illustrate this with water. You have pools and metabolites reactions contributes to different metabolite pools in multiple ways. And overall, when we impose this steady state, all we're saying is the rate of production and the rate of consumption have to balance each other. And this leads to these very nice linear equations where the variables are the rates or fluxes of the reaction. So hidden inside each of these reaction rates is the whole Michaelis-Menten kinetics. But somehow, when we go to this steady state approximation, we just look at the steady state rates of the reactions, which makes for much simpler and more tractable systems. Of course, we somehow give up understanding the steady state concentration of metabolites in the same way as from knowing the rates of water going in and out of a pool. At steady state, you wouldn't be able to tell the amount of water that is in the pool. So that is actually really important to keep in mind. So when you represent a reaction network to write these types of equations, this is nicely represented in the form of what is called a stoichiometric matrix, where each column represents a reaction and tells you how much of each metabolite is consumed and produced. And this is a very elegant and useful representation of metabolism. This is the stoichiometric matrix for this very small network. If you look at larger networks, this is the same example you saw before. This is our simplified version of this heterotrophic bacterium. And this very complex diagram, which you can find in this review article written by Helen Scott, illustrates the fact that in addition to the reaction stoichiometry, you need to know in order to solve a flux balance problem, you have to have information about how much of each metabolite is available from the environment. You may have information about genes associated with individual reactions and potentially multi gene to reaction mapping, as multiple genes may form multi-protein complexes that are effectively the enzymes. So the advantage, once you solve this problem, and for example, one can use linear optimization to find the set of fluxes that maximizes production of the biomass, finds the most efficient way for cells to grow. This is, sorry, the same stoichiometric matrix for this slightly more complicated network. So you can imagine how messy this becomes for real networks, but it is a matrix, right? It's a formal representation of metabolism. This is actually very important. One can solve this problem and ask questions such as, given a certain amount of glucose and oxygen coming in, what is the maximal amount of biomass that can be produced? And you can easily change the constraints. For example, remove oxygen from the model, and now you'll find that the organism will ferment, produce some fermentation byproduct, and produce a smaller amount of biomass. You can change the objective function. Often, flux balance analysis is presented as a method for maximizing the biomass production, and that's usually how it's used, but it's not the only way to use flux balance models. For example, in metabolic engineering, where this method originated, one can ask what is the ideal maximal yield of producing a certain metabolite, for example, this molecule A. So the encoding of information in flux balance models is a part of the question. The other question is how to use it to produce different phenotypic predictions. Now, the construction of models from genomes, and many of you are familiar with this, is a whole field now in itself, right? One needs to go from the collection of all known metabolic reactions across all living systems through information about the specific reactions found in the genome of the organism of interest, often through the usage of literature curation. The ultimate goal for making predictions for a specific organism is to build this, what is called network reconstruction for a specific organism. This is E. coli from EcoPsych, and this has the formal representation of the stoichiometric matrix. This process of going from genome to network is actually quite complex. There are many tools that can be used for this. KBase, the Department of Energy Systems Biology knowledge base, has multiple tools for this, but there is a whole field now of different tools for creating draft networks, trying to fill gaps in metabolism. And one thing I want to point out here is that this is an imperfect process. It's very difficult to make models, especially for newly sequenced organisms. And I think this is still very important. And even if we have imperfect models, it's actually quite instructive to try and build models because it illustrates that it's even just formally keeping track of what we know and we don't know is very difficult, but it is very important, irrespective of whether or not these models are used for different types of quantitative predictions. So the process of building these models is interesting and important. And of course, ultimately, one wants to use these models for making predictions that are relevant for different fields, including marine biogeochemistry. This method, known as flux balance analysis, really has its roots in metabolic engineering in the 80s. And one component that is also very important in making predictions is the biomass composition of the organisms, which is the ultimate sink of metabolites in the cell. This needs to be measured experimentally. This is the biomass composition of E. coli, the different amino acids, phospholipids, and so on. And one thing that is still part of ongoing research, and people are trying to explore this, and I think links nicely with models, for example, of macromolecular compositions, is that the composition of a cell is dynamic, and we usually in flux balance models take a fixed biomass composition, but I think there is a whole open field in trying to make this biomass composition dynamically changing as a function of environments and so on. The other thing I want to point out for, as one application that we're actively working on in the lab for flux balance modeling is the estimation of carbon use efficiency. We did prior work on a number of organisms with automatically reconstructed models. We're now in the process of evaluating in more detail how carbon use efficiency, the ratio of biomass that goes into biomass versus the carbon that is uptaken by the cell, how does this carbon use efficiency depend on different parameters, including the nature and composition of the carbon, the maintenance, interaction with other organisms, and so on. I will, before going into how to extend models from organisms to ecosystems, I want to point out, summarize some of the pros and cons of this method of flux balance modeling. As I said, this is a good representation of knowledge. It's very fast. Those of you that haven't tried it, it may seem very complicated and very time consuming, but flux balance modeling has the advantage of being very fast. It takes a fraction of a second to predict all reaction rates in a cell. There are no kinetic parameters, and we care about the fluxes. As I mentioned, this is limited by knowledge, so gaps in the network and missing genomic information is an ongoing challenge. It's very difficult to predict metabolite concentrations and intracellular dynamics. And one of the ongoing challenges is also incorporating regulation and signaling. So marrying genome scale models with omics data is obviously something that many people are interested in, but is also very challenging because of the nonlinear nature of the Michaelis-Menten kinetics that connects concentrations to fluxes. But I want to jump into what I think is the frontier of this field, which is trying to go from single organisms to communities. And we and others have been thinking about this in different ways. And I want to just give you first an overview of different ways of extending genome scale models to communities. In the same way as you build a stoichiometric matrix for a single organism representing metabolism of that organism, one could build a stoichiometric matrix of a collection of organisms where you keep track of the metabolites present in different organisms. This is, again, a soup of enzymes approach where it's as if you assume that the boundaries across species don't matter, and whether or not that's true, that's an open question. A type of approach that have been very useful and is still being used a lot is this multi-compartment model, where you can imagine the collection of multiple species being put together into a single block stoichiometry. Essentially, this is treated as if the two organisms are different compartments in a cell, and this makes it feasible to construct communities of even high complexity, more than two species. The problem with this type of approach is that it is very difficult to predict the abundance of different organisms using this kind of multi-compartment model, for the same reason that we cannot predict concentration of metabolites in the cell. The other approach, which I'm going to dive into, is using dynamic flux balance analysis, and this will keep the two organisms separated, but allow you to predict, as you'll see in a second, ecosystem level concentrations and dynamics. I'll mention later these consumer resource models. So the idea of dynamic flux balance analysis is the following. You can take, again, a genome scale model that predicts growth as the biomass production of the cell. And now you can start with an initial concentration of environmental metabolites, say glucose, and an initial abundance of a given species. And you can predict at this time, using flux balance analysis, you can predict the rate of growth, which essentially is the slope, initial slope of this growth curve. And you can predict also the rate of consumption of a metabolite. This is the flux of nutrient utilization. And in order to translate concentration into flux, you need a Michaelis-Menten For the uptake rates, so the dynamic flux balance approach is a hybrid approach that treats intercellular metabolism as a steady state, but does use kinetic equations for the uptake rates. So now once you have this prediction of the initial usage of metabolite and growth rate, you can extrapolate this for a certain period of time. This should be a very short time. How short exactly is something that can be addressed in different ways. But essentially, this is equivalent to a finite difference approximation where you can now uh, extend the time piecewise approximation to multiple time points and calculate effectively the consumption of a metabolite, the production of biomass, create a curve that looks like a growth curve and utilization of resources. Now, in addition to the fact that this allows you to potentially look at dynamical processes such as dioxic shift and so on, dynamic flux balance analysis is very helpful for looking at communities. And I should point out, this method was published originally in 2002. It's a beautiful paper, and I think only later on it became a very prominent method for community dynamics. And one of the reasons is the following. Imagine now as this organism is growing, you can simulate and generate in flux balance the flux of production of a metabolite that is secreted, for example, acetate, and this will accumulate in the environment. And now if there is a second organism, this yellow one, that could not grow on glucose but can grow on acetate, and if you just iterate over flux balance calculations for the different organisms, you'll see this yellow organism will start growing because of the acetate that has been secreted. And what is nice here is that you can effectively simulate an exchange and interactions between different species while still assuming that each organism maximizes what is good for itself. There is no ecosystem level optimality criterion to be invented or suggested. You can still assume that each organism is maximizing its own biomass, for example, and the interactions between species, both competition and cooperation, will emerge as an emergent property of each organism doing its own metabolism. So this is why dynamic flux balance analysis, in my mind, is quite appealing for modeling ecosystems. In the past, we've applied dynamic flux balance modeling to the growth of Prochlorococcus. This was an effort with Daniel Scher and others. And what is nice is that it's possible to include light in the modeling, and you can simulate day-night cycles, and correspondingly, you'll see the biomass production having cycles of growth and resting, and the glycogen changing over time in a periodic manner, matching the DL cycle. I should say this work was a combination of revisiting the Prochlorococcus model that had been built by John Casey and others before, and also imposing flux constraints for the uptake of carbon and light in the Prochlorococcus model in a way that we called push FBA as opposed to the standard constraints because the control of light input and CO2 input in this model is different than the transport control input of how usually flux balance models are built. I'm not going to have time to go more into this, but you can find out more in this work on this paper. I want to show how we extended this idea of dynamic flux balance modeling to also include the spatial structure. And the idea is the following. You can look at a box of space and assume that there is a certain concentration of microbes in that box. In that box, dynamic flux balance analysis will operate as I just illustrated. And in addition to this, you can model diffusion of molecules and cells across a grid. Ultimately, developing simulations that will take into account whole cell metabolism as well as the growth process and diffusion of molecules. This is a simulation of E. coli on a Petri dish. And this is what we call COMETs, computation of microbial ecosystems in time and space. It started as a software for modeling ecosystem dynamics. I'm realizing I'm out of time probably, right? So I should wrap up soon. I apologize. I will just mention Comets is free and available, and it's an open source software that I hope people will have a chance to use. I will just jump to a few concluding thoughts on how to actually bridge the scale. So one way, I think one avenue that will be very important is connecting models of different scale. This is something we're actively exploring. CComp, kind of plug and play, ways of having plug and play between models, coarse graining, genome scale models, I think can be coarse grained to less detailed models, including consumer resource models, where there is very little detail, but one can extend to many, many species. Integration of AI with mechanistic models, especially phenotypic data, I think has a lot of importance. And finally, theory. And again, I don't have time to go into this, but this is from an article with Mick Follows and Daniel Scher, showing how there are basically conserved laws across scales. And I think this will be very important as a component of future efforts. Thank you for listening. Thanks to collaborators and funding agencies and stuff.
Thank you. Thank you very much. That was fantastic to me. So questions. Maybe Charlotte, I can give you the mic if you want to ask your question. If you could, maybe it's easier for you. And then I will.
Okay, yes, sure. I wasn't prepared for that. Okay, so great talk. I really liked it. So I was thinking that the large biogeochemistry models that I usually use, they do not account for heat stress in plankton. So if the water gets warmer, the plankton just grows faster. And the underlying assumption is that simply a different species would take over that has a higher heat tolerance. And so when you then have climate change in those models, you usually get a shift towards smaller plankton types to alleviate the nutrient limitation. But there is no trade-off regarding the heat stress. So the problem that you described, that you actually also want the larger cell to have some heat protection, is not included in our models. I don't know if you have an idea how this could be improved or have something to say regarding this.
Yes, I do, because that's really the next step where I want to go next. Collaborate with Earth system modelers to try to implement some of these insights that we get from protein modeling, right? So I believe the way to go is not like plugging a whole protein model into a large-scale biogeochemical model. For sure, that's not what I suggest, but I believe we can like learn from these protein models what are the trade-offs that matter, right? At least under this simplified approach that we are thinking about a single cell maximizing their growth. And I think this is very possible in a way and not in a very complicated way. If we think about it, there are some models already, biogeochemical models, that represent the full thermal curve, the unimodal response of growth as a function of temperature. And this is maybe a first step so that we can account for this decay in growth, right, under higher temperatures. But more importantly, perhaps, is to think about how we define trade-offs in biogeochemical models. So you were saying about nutrient uptake efficiency, right? So we have an allometry there where we defined nutrient affinity as a function of cell size. And we know that small cells have higher affinity. So we could perhaps think about thermal tolerance in a similar way and get the insights from the protein model to derive these allometries, right? So if we plug in now this other trade-off in biogeochemical models, what would happen? So something that I am interested about is like, would this advantage to the larger, because they are more heat tolerant, really give them space within a model that can account for ecological interactions, predation, and other dynamics? So this would be very nice to explore. So I think this is one potential way that we could go forward. But of course, like getting more empirical data to also derive this allometry would be very important.
And there is two questions that I'd like to put together for Daniel. One question from Xenia, which is how the biomass reaction is adjusted for each species. And it's maybe linked to the question from Adam, which is about, you mentioned, Daniel, the major gaps in developing cellular scales models. Could you speculate and discuss what are these knowledge gaps? I think it's linked to the first question as well. So
Yeah. So the biomass, I mean, in a lot of the literature, there is a gram positive and a gram negative biomass, and a lot of the automated tools just use gram positive, gram negative. Some individual papers go into trying to find the specific lipids, specific compounds that are produced just by individual organisms, but that needs typically experimental data. How important this information is depends probably on the questions you're asking for a model. If you're asking about, fundamental energetics or overall growth rates, may not matter so much. But if you're interested in, let's say, phosphate limitation and an organism produces phospholipids under phosphate limitations, those lipids have to be added to the biomass based on knowledge of that specific organism. So definitely the biomass composition is probably an important component of the models that we often don't know. I think the most challenging part is that, many genes have no known functions. And when, the way I think about this, when you build a model, it's as if you have a city, but there are whole streets that are not there, and you cannot go from one place to another. And that's the big challenge. Sometimes, you can add streets based on, knowledge of other organisms, but doing that well, I think, is extremely challenging and requires good algorithms and which people are developing. So I think genome annotation is still one of the limiting factors.
And I have another question. It's maybe linked to the Suzana and Daniel. Suzana, you mentioned the question of temperature, which is something that I did not see from Daniel's talk. So do I understand that if I need to tackle the question of temperatures, I need to push more for proteomes models? Or Daniel, how do you think that you could handle the temperature, which is actually a big deal in ocean studies?
Suzana, you want to go first?
I think you can start. Yes.
Okay. I'll just say, I mean, in flux balance models, there is really no temperature, which it's been a challenge. I know of a few attempts. I think there's been, you can in principle use Arrhenius equation to modulate fluxes, but there are so many factors that are temperature dependent that could affect fluxes, and I don't know that we know how to do that mechanistically. I think another idea I think I've heard floating, and is to add, you can add a modulating curve, temperature dependent curve on top of the model, but that would have to be measured experimentally. I don't know that models right now can, from first principles, predict temperature dependence.
Yeah, and maybe I can just add to that, like within the protein model, we represent metabolism in a coarse way, right? So we have a few reactions, and we need to define, as Daniel was saying, like what is this thermal dependency? How fast are they increasing as a function of temperature, these different reaction rates? And we can use different thermal dependencies if we know the information. So for me, that I have less pathways in the model is not as challenging because we know, for example, photosynthesis should be less thermal dependent or independent of temperature. So we can define that, and this really helps to make predictions. So yeah, I think that's my contribution to this question.
Okay, and I have another question that actually for Daniel, but I think it could be for you also, Suzana. So I'm sorry I distribute the question like this. It's about the experiments, and do you think there is a way that we could determine the parameters that are really the most important to measure in order to maximize the model improvement? It's like in your models, are the parameters all equivalent, or do you think you could identify the most important one in a way that maybe to help experiments and to move forward?
Yeah. That's a great question. I mean, the stoichiometric coefficients, I assume we don't think of those as parameters, or those are the stoichiometries. Although, yeah, one could try and understand what stoichiometric coefficients the model is most sensitive to. I think the most important parameters that we'll have to address for community models are uptake kinetic parameters. And I think a way to do this would be to do sensitivity analysis. And we know that often if a nutrient is not limiting under certain conditions, of course, the kinetic parameters don't matter that much. So yeah, I think that measuring um, yeah, performing sensitivity analysis is one way of determining which parameters are most relevant to measure. I don't know if that answers your question.
Yes, I agree with Daniel. So we would take a similar approach, performing sensitivity analysis. And by running the model, you can identify, right, which like parts we have more uncertainties and that we should focus and get data for that. So I have been doing this proteome modeling work made me look a lot at the literature of people that actually have measured these thermal curves under different conditions. And there is some data, but not a lot. So it's hard to find data for different traits. So this is something very important. Like the more you can describe phytoplankton physiology as a function of these drivers that we are interested, the more we will be able to validate our models with more confidence because there are a lot of parameters. So that's something important to take into account.
Okay, thank you. Naomi, I give you the floor to
All right.
conclude. Into exciting news.
Well, thank you again, Daniel and Suzana, for such awesome talks. I've personally really loved all three webinars. And just a reminder that there'll be an in-person workshop. The applications are open now. We're really excited to get everyone together into the same room and continue some of these discussions we've been having. And I also really wanted to thank the co-organizers of both the webinar and the in-person. Damien, Charlotte, Christoph, Al, and Sophie have put in a huge amount of time. And the in-person workshop is going to be co-sponsored by CComp that Daniel mentioned. So it's going to be a really great opportunity to bring lots of different ideas and people together. And so with that, I think we'll end the third webinar, and hopefully we'll see a good number of you in September.